don't have screen sharing.
Okay, we can fix that for you.
Okay.
Sarah's going to
Okay,
fix
I
that for you.
I have it now. Okay. I hope you guys can all see and hear me clearly. And before I even start, I'm going to apologize if there is any issues with the connection. I'm in India right now, so the internet is not always very reliable. Thank you everybody for joining to hear me out today, and thank you Liz and Scott, Liz and Jay, and everybody at the Alex's Lemonade stand for giving me this opportunity to present our work. We all have heard a lot about immunotherapy the past few times, or quite a few times in this series, as well as in various conferences these days. And I'm going to turn it around and talk about a little about macrophage immunotherapy, obviously distinct from T-cell immunotherapy. A little bit of this was introduced very elegantly by Robbie Masner earlier on in this series, and I'm going to expand a little bit more on what we have been doing in our lab and why macrophage immunotherapy. Now, immunotherapy in general is based upon or based around the cancer immunity cycle, which basically starts here at stage one, which starts off by the release of cancer cell antigens, which is released by the tumor cells, taken up by the APCs, eventually trafficked into the lymph nodes, presented to the T cells, which then become tumor-specific T cells, which eventually, in theory, can attack and kill your tumor cells. Now, most immunotherapy is based upon the T cell world or around the T cell world, essentially the checkpoint inhibitors, PD-1, PD-L1, and CTLA-4. What we are trying to do is to see, can we really modulate the macrophage compartment within the tumors itself, and which come from the periphery, into working for us in that can we use macrophage immunotherapy to attack tumors. Now, one of the biggest problems for brain tumors, especially pediatric brain tumors, when it comes to immunotherapy has been the fact that it has a very low mutational burden. And essentially in this graph, you see most of the pediatric tumors towards the left side of the graph. What I'm going to talk about, like I said, is the macrophage immunotherapy and a central phagocytosis checkpoint called CD47, which eventually allows the macrophages to go and start eating the cell. Now, this taken from, and I apologize, I forgot to put in the reference for it, is a very elegant diagram which not only talks about the macrophage functions, but also the various strategies which are using for targeting macrophages for tumor immunotherapy. Now, macrophages more and more are turning out to be a very key player or a central player in both tumorigenesis as well as immune suppression. But not only that, we now know that they play key roles in both radiation resistance, chemo resistance, as well as resistance to immunotherapy. Most of the functions of the macrophages, when it normally comes to tumorigenesis, arise from their function as secretion of various cytokines. So you have the tumor promoting functions by secreting various inflammatory cytokines which lead to tumor growth. They also suppress their own function by, no, sorry, I apologize. They don't, they also function, they become polarized towards what we call as M2 macrophages, which is not exactly very distinct from M1, as we now have come to know from all the beautiful single cell data that is coming out. But these tend to be very immunosuppressive, prevent T cells from infiltrating in there, and promote tumor growth eventually. So the various ways that they're using or people are targeting macrophages has been by inhibiting the tumor promoting function or directly actually blocking all the secretion that occurs, or suppressing the recruitment of macrophages for example, by targeting the CCR2 pathway. There is also various ways by using CSF1 inhibitor, which reduces the survival of the macrophages. But what I'm going to mostly talk about today are two major points. One is removing this phagocytosis blockade or phagocytosis checkpoint blockade by using reagents which target the CD47 SERP alpha pathway. But something that we have now started working in the lab, and that is trying to use chimeric antigen receptor expressing macrophages, which modifies these effector cells or modifies these macrophages into directly targeting the tumor cells by essentially eating them up. Now, the phagocytosis checkpoint essentially works in the following way. Well, so we have CD47, which acts as a don't eat me signal, and that binds to SERP alpha. This leads to an inhibitory signal which prevents phagocytosis from occurring. If you block the CD47 SERP alpha interaction from occurring, this will now activate phagocytosis and induce the eating up of the tumor cells. One thing to keep in mind is that not only do you need the blockage of the don't eat me signal, but you actually need an activating ligand which acts as an eat me signal into binding to an activating receptor on the macrophages as well. So in a sense, oops, sorry. So in a sense, it is this balance between the eat me and don't eat me signal which is really required to induce this phagocytosis to occur. Now, most cancer cells lack the inhibitory antigen, and if you have the innate immune cell, this will inhibit the macrophage from going and eating the cells. If you use drugs to block this inhibitory signal, you can then use either you use the drug to block the inhibitory signal, but you still, sorry, phagocytosis activating signal, sorry, uh, and then you still need to get rid of this inhibitory signal, which can again be either an antibody, which itself has an FC portion, which acts as the effector portion of the antibody, in initiating the macrophage from going in and eating the cell, or you can use drugs to do the same effect, essentially activating these eat me signals on the tumor cells, which eventually allow the macrophages to go in and eat them. So the take-home lesson at the end being you need this activation of the eat me signal, whereas at the same time blocking the don't eat me signal. We started this work and I got involved, this is back in Irv Weissman's lab. And essentially what we had done was we threw in macrophages along with tumor cells, tumor cells being in green and the macrophages being in red. You add in the anti-CD47 antibody and you can actually see these macrophages literally going in and engulfing the tumor cells. The thing that I really want to point out here is the presence of this NSC1 and NSC2. These are human neural stem cells. And the thing to point out here is that even in the presence of the CD47 antibody, you do not have phagocytosis of the normal neural stem cells. Our initial studies was using a commercially available anti-CD47 antibody, which we then developed a humanized anti-CD47 antibody, HU5F9G4, and it's now called magrolimab, and is in clinical trials being run by Gilead. But this particular engineered antibody had an IgG4 variant, which was used to reduce the FC function. And we went on to show that it has all these other characteristics. And when I say we went on to show, this was mainly in non-CNS solid tumors as well as non-CNS hematopoietic tumors. So initial studies were done in adult glioblastoma. This is a primary patient GBM. We orthotopically implant them into mice brain, which gives this massive spread in the brain itself, crossing the corpus callosum and pushing against the midline. you start treating them with anti-CD47, you clear them out. We have also done this in a spontaneous breast cancer model where we actually established the tumor in the fat pad of the mice. We then went on to resect out the primary tumor and let the mice live. They not only gave rise to beautiful these lung metastases or these nasty lung metastases rather, but they also gave rise to these micrometastasis within the brain of the mice. And when you treat them with the CD47 antibody, they would clear them out. Now, our initial studies were done in about 5 different pediatric brain tumors, including DIPG, and I'll show you small vignettes from them. This is work that has already been published, which has already been published, so it's easy to look it up. Essentially, we do the same thing. We inject the cells, we would randomize them and start treating them, and you follow them by imaging. You see a suppression of the clearance of the tumor, rather increase in survival, and especially if you image the mice or image the brain, you could clearly see complete removal of all the tumor in the hindbrain itself. One of the things that we really wanted to focus on was spinal metastasis, or rather leptomeningeal metastasis, mainly because, especially in MYC amplified medulloblastoma, that is the main cause of morbidity in these patients. We developed a line from a C-Mye amplified medulloblastoma, which not only gave rise to which not only gave rise to spinal tumors, but it also gave rise to this beautiful leptomeningeal spread all over the brain. And treating them with the CD47 antibody clears out not only the primary site, but also clears out the spinal metastasis. We then went on to see whether we could actually treat this by intraventricular administration of 5F9G4. And in this case, what we would do is, instead of doing an intraperitoneal injection treatment, we put them into osmotic pumps and directly infuse them into the brain. And we see very similar results where we see not only we see clearance of the tumor, but also an increase in survival, followed by an influx of macrophages within the tumor site. The one thing that we really wanted to see was there an additive, was there an additional effect of infusing the antibody versus giving it intraventricular? And sure enough, what we actually see is an accelerated effect of the antibody in clearing out the spinal tumors as compared to systemic treatment of the mice. One of the things we really wanted to focus on at this point, or at that point, had also been whether or not there was any toxicity on normal human cells, keeping in mind that this particular antibody does not bind to any mouse CD47, it only binds to human CD47. So what we did was we took normal human neural precursor cells, labeled them with a red dye, took tumor cells, labeled them with a green dye, mixed them up together, mixed them up with macrophages, with and without the antibody. And then we went on to see whether or not it would take up both human tumor cells or the normal cells as well. And what we really see is that it preferentially phagocytoses tumor cells and not the normal cells. We went on to do this in various different normal lines with the same effect, including differentiated neurons as well as astrocytes. We went on to do an in vivo evaluation of the toxicity on normal human cells. In this particular experiment, what we did was we took the normal human precursor cells and injected them into mice brain, and then into mice pups. We isolated out those pups which had engrafted human normal precursor cells, which we see by luciferase imaging. In this case, the luciferase imaging is coming from the normal cells and not from the tumor cells. And what we see, and then we started treating them with either control or 5F9G4, and we see that it did not affect the It did not affect the normal neural precursor cells, and but it also increased survival, something that I've not shown here. So it did clear out the tumor cells, but had no effect on the normal cells. Um, a very qualitative analysis was carried out as well, just to see what the normal behavior of the mice looked like. Clearly seeing on the left-hand side is the control, on the right-hand side is the mice treated with the CD47 antibody. And we see the mice on the left all hunched up, they were cachectic, fur was all messed up, whereas the ones on the right were healthy, roaming around and jumping around. So it had a very clear qualitative effect as well on mice behavior. We have gone on to show this in an immune competent model as well, using an anti-mouse CD47 antibody. Now, this particular antibody, or rather, I should say that there are actually not very good anti-mouse CD47 antibodies, so it tends to work not as well. But still, giving fairly high doses of the mouse CD47 antibody, we still saw the same effect, including an increase in survival as well as an influx of macrophages. So another thing that we wanted to see was that, well, we do know that there is already a resident population of macrophages in the brain known as the microglia. And this was work done by Gregor Hutter, Johanna Theruat, and Klaus when we were in Sam Cheshire's lab at Stanford. And we went on to use a mouse model which allows us to color code the macrophages and differentiate them from the microglia by using the CX3CR1 GFP, which is actually marks the microglia, and CCR2 RFP, which marks the macrophages. And you can see here that in the contralateral hemisphere, you primarily see only microglial cells, whereas in the tumor site, you see both microglia as well as macrophages. Now, what we did was that we backcrossed this mouse into the North-Skid gamma immune compromised mouse, which now allows us to have this color-coded system and be able to implant xenografts into them. So the first thing that we see is that so one of the tricks that we can use with this particular mouse is that if you have the CCR2 double knockout or the homozygous knockout, you do not get any infiltration of macrophages. Well, to correct this, I mean, you get very limited infiltration of macrophages into the mouse brain. And what we really see is that even when we had blocked out the entry of macrophages into the mouse brain, we still see an effect of the CD47 antibody, strongly suggesting to us that the microglia also were capable of acting as effector cells when the CD47 phagocytosis checkpoint is blocked. The thing that was very interesting was that even in the CCR2 knockout tumors, we did get a decreased T cell infiltration. The CD3s were all over the place when you actually knock out the CCR2 knockout, but if you specifically start looking at the CD4 or CD8 T cells, you actually start looking at fairly decreased numbers of T cells. And this is a mechanism that we have not explored yet, but something which is on our plate right now. But we really wanted to see what really happens within the tumor bed itself. So we moved to a technique called live animal 2-photon intracranial imaging, which is essentially a technique where you take the skull of the mouse, you remove the skull, you slap on a window, and now you can actually do fluorescence, 2-photon fluorescence imaging to look at the dynamics of macrophage and microglia interaction within the tumor bed. Now, shown here in green is the microglia, and the red spots are actually monocytes coming in. The blue obviously is a blood vessel, and this is within the tumor bed itself. Even though the tumor is actually color-coded as well, we don't show it up. And this is in pseudocolor, so we can see the blood vessels very clearly. what we end up doing. And if you don't give CD47 antibody, you see some amount of movement occurring in the microglia, but fairly, you see these monocytes in the blood vessels itself, and every now and then you will see one of them extravasating into the tumor bed itself. But when you give the anti-CD47 antibody, you see this massive influx of monocytes coming in and infiltrating into the tumor bed, clearly showing a peripheral recruitment of the macrophages. But what really happens to the microglia itself? So we ended up rendering these images when we imaged them for a long time, or we took a long time video of what was happening. We do not have macrophages in here. And right now what we are seeing is the microglia sending out these long processes and surveilling the surface of the tumor cells and eventually engulfing them or phagocytosing them. So microglia are very active. They're actively surveilling the surface of the tumor cells, and they're also capable of phagocytosis. So, so now switching gears a little bit, I'm going to talk about one of the ideas that we are now working towards is, and not just us, a lot of labs are doing this, is how can we really make a cold tumor hot? Now, uh, the standard of care, that is chemotherapy or radiation therapy, can have two different effects depending on the reagent that you use or the radiation modality that you are giving them. It can be an immunogenic cell death or a non-immunogenic cell death, and the non-immunogenic cell death actually giving rise to what is known as tolerogenic phagocytosis, uh, which actually leads to immune suppression. It increases the Treg activities and decreases your cytotoxic T cell responses, and eventually leads to tumor growth. Whereas your immunogenic cell death can, as the name suggests, lead to an increase in CTL response and overall tumor destruction. So trying to look at the various modalities which can shift your cell death towards more of an immunogenic cell death is one of the goals of the lab. How do we do this? We actually do this by, sorry, we actually do this by increasing what is known as damage-associated monocular patterns on the surface of the tumor cells. We know that there is a translocation of calreticulin on the cell surface. There is also HMGB1 release as well as the ATP release occurring, which eventually attracts both the macrophages into the tumor bed and leads to the eat me signal upregulation, eventually leads to phagocytosis of the tumor cells. We've shown this with radiation. I apologize, this is not D425, this is actually an adult glioma. That when you give radiation, you actually can enhance the phagocytosis of CD47. This was work done in collaboration with Sam Chesh's lab. And we actually see a decrease in tumor growth when you combine radiation with CD47. Senthil Chetty in the lab has now taken this and started working on diffused midline gliomas to see whether we can use the same modality in the pons of these mice to see if radiation can combine with CD47 therapy. And sure enough, even at high tumor burdens, when you combine fractionated radiation with just 1 week of CD47 treatment, you start seeing a significant decrease in the tumor volume. And this has been taken out for more than a week now, and you start seeing an increase in survival as well, shown here. He's done it with a cell line called BT245, which is a thalamic GBM. I apologize, I don't know why these are squished together. But we have seen this in both BT245, which is a thalamic GBM, as well as DIPG 17, where we see an increase in survival when you combine radiation therapy with CD47. We have also looked at the phagocytosis of DIPG17 cells upon fractionated radiation, and once again we clearly see an increase when you combine fractionated radiation with CD47. And Senthil has gone on to show that you do see an increase, a significant increase in calreticulin on the surface of these DIPG cells. One thing we wanted to check is what happens if we actually give sublethal doses of chemotherapy. And this is basically meaning that we are giving doses where you do not really see significant cell death, but using a very commonly used chemotherapy regimen of CCNU, uh, vincristine, and cisplatin, we actually see that even at sublethal doses, you can increase phagocytosis when you combine with CD47. Now, at this point, we have to really go back and look whether this is happening from an immunogenic cell death perspective or this is simply efferocytosis. Because one thing now we need to keep in mind is that if it's simply efferocytosis, we could actually be inducing immune suppression. This is something which is ongoing in the lab right now. But now we have to make that distinction whether this phagocytosis is now occurring from efferocytosis as just live cell phagocytosis. Um, a study that we carried out or have been actively carrying out with Mark Remke's lab in Dusseldorf has been the epigenetic regulation of immune evasion in MYC-driven medulloblastoma. Uh, what we essentially did, this is a manuscript which is now under review, is that Mark ended up looking at a whole bunch of FDA-approved reagents and then separating them out based on those which had very high activity against MYC amplified medullo specifically. So everything which is below the group 2 and group 3 was discarded because these had very high IC50s. And we started focusing on those which were really had these low IC50s and those which specifically had low IC50 for MYC medullos. And one of them that we came out with was CI994, which is a HDAC inhibitor. Now, CI994 has very nice specificity for MYC medulloblastoma compared to GBMs, ATRTs, or other medullos. Seen here looking at the IC50, this was work done by Victoria Marquardt and Joanna Theruwath in the lab at that time. CI994 induces apoptosis, and in fact, it actually downregulates levels of MYC in the treated cells. But more importantly, by using this UW228 line, which had low MYC to start off with, if you overexpress MYC in it, you actually increase the sensitivity of these cell lines with to CI994. And you do get some level of single agent activity even in the mice. However, our interest really came in when we started looking when we started running RNA sequencing and methylome analysis at low doses of CI994 on the MYC amplified cells. And to our surprise, the top hits which started getting pulled out were those which were associated with innate immune activation. So you had your upregulation of TLR4, interferon alpha 2, NF kappa B, TGA beta 1, and interferon gamma. So this really led us to think whether we can really start using this as a mechanism or as a way to tease out how immune evasion works in MYC amplified medulloblastoma. And we went on to do a preclinical analysis to see whether it really combines with CD47. And sure enough, not only it increases the phagocytosis of CD47 at low doses, but it actually significantly enhances the survival in these MYC-driven medulloblastoma xenograft mice. This is a study which is still ongoing in the lab, but just looking at this sensitizing or immunizing the tumor cells, A couple of things that we have seen is that if you look at patient samples and look at the surface expression of CD47 on some of the patient samples that we got, you actually see an increase in CD47 expression in the group 3, that is the MYC amplified medulloblastoma lines as compared to the non-group 3 lines. We have seen this both in a allograft mouse model, one which was developed by Bob Rexlarea, the MYC-GFI P53 model, and especially when you compare to two different sonic hedgehog mouse models, you clearly see an increase in CD47 expression on the surface. We have also looking at a new reagent that is a direct MYC inhibitor, that is MYC-CI975, which shows exquisite sensitivity to D425, which is a heavily MYC amplified line. And not only this, this actually starts inflaming the tumors, that is increasing NF-kappa B expression when you treat MYC amplified lines with the MYC-CI975. We have to obviously go back and do all the controls and knock down MYC and do all the experiments there, but this is extremely telling that just modulating MYC levels, you can actually start going after and starting to combine them with CD47. There are obvious links to the adaptive immune system, which is ultimately where everybody wants to go to. Can you really drive the adaptive immune system? So the answer to that is yes. However, we have to be very careful on how we are driving this. This is something that we really wanted to see. Can we combine CD47 with PD1? But the take-home lesson from this slide is that you cannot just throw them together. That is, you cannot just take a tumor and throw both CD47 and PD1 together. You actually, if you combine them, you do not get any effect which is more than CD47 alone. You have to start treating them sequentially, meaning that you first give CD47 for a couple of weeks, you wait for a washout period, and then go back and give PD1. And that is when you really start getting this increased effect or increased survival in these mice. So timing is just as important as the reagent that you're using. And lastly, not but least, we are also starting to look at chimeric antigen receptor expressing macrophages. We have all heard a lot about CAR T cells and their all the fascinating work that has been going around it. But we wanted to really see that can we make macrophages function and behave in the same way as CAR T cells, that is essentially providing them with a homing mechanism, homing and activating mechanism, which can make the macrophages go into the tumor cells and eat them. And why we really why we are focused on CAR macrophages is that macrophages or monocytes especially are designed to infiltrate into solid tumors as compared to T cells. And so this is just a proof of concept that we very recently did in the lab. And this was a combined effort from Eric Hoffmeyer, Alison Cole, and Senthil. And what they essentially did, so in this case, we are using Raji cells and we are using the very standard CD19 CAR, which has been very well tested a number of times now. And so we made these macrophages with the CD19 CAR and fed them Raji cells. In this case, do keep in mind, I'm using GBM as a control because GBMs normally do not express CD19 on the surface. This is slightly opposite to what I've been talking about till now. The Raji cells are the target cells here. And what we really did see is that if you look at the blue and the green, that's just Raji cells with just macrophages, whereas the blue is the GBM cells with the CD19 CAR. They both don't respond because one doesn't have the CAR on it and the other one doesn't have the target on it. You add the GBM with, even with the CD19 CAR and CD47, you do see an increase in phagocytosis. But more interestingly, the Raji cells really get very rapidly eaten up But what really surprised us was this massive increase in phagocytosis when you have both the CD19 CAR as well as the anti-CD47 antibody. So this is a proof of concept to show that macrophages can actually go, and this is nothing new. Also, I must admit, Charisma Therapeutics, there's a whole company behind it coming out of Sargel's lab, as well as some really beautiful work which has been done by Courtney Crane at Seattle Children's. And this is something that we have now picked up to see whether we can use these in pediatric brain tumors. So the one big question comes is that are all macrophages equal when it comes to eating? This is again something very new that we have started in the lab. We really wanted to see. So one thing that we have always seen and noticed is that there is always a threshold of how many macrophages we can really get to eat tumor cells. And depending on the donor from where we get those macrophages, it can range anywhere between 20 to 30% at best. And that is something which we have tried over and over again. No matter what we try, we can never make more than 20% of these macrophages actually start eating. Even though the way they are cultured and the way they are isolated, it's all the same, only about 20%. And it's been in culture for anywhere between 7 to 10 days. It never really goes beyond that. So we wanted to see what was really this difference between eaters and non-eaters. And so what we did was that we started the macrophages, we combined the macrophages with tumor cells, and we let them go for 24 hours, and then Senthil sorted these double positive, which we labeled them as eaters, and the single positives, which was the non-eaters, and then we did single cell RNA-seq on it. And we're still deconvoluting this data to figure out, but couple of things which already came out. Number one, there are actually two very distinct clusters which come up, two being the eaters and one being the non-eaters. You can see the eaters separating out from the non-eaters. But there was also very differential gene expression occurring. These are the top hits. And this is just the very surface of this massive gene list that has been created now. That there are actually differences in what are the top genes, whether when you treat them with CD47 and when you don't. So clearly there is this heterogeneity which occurs not only in the macrophage population, but also in the actual functional phagocytosis or post-phagocytosis within the macrophages as well. And just to give you a flavor of it, in the non-eaters, you still have cytokine signaling going on, you have leukocyte activation going on, including interferon gamma mediated signaling. But clearly in the eaters, you have there's a huge increase in the lysosome genes as well as the phagosome genes. But what we have also seen did not really show up here, but we do see some increase in antigen presentation genes also occurring. So this is something we hope to be able to present to you down in the future, sometime in the future. So that is pretty much the end. All the 47 story actually started out in Irv Weissman's lab, and when I was working with Sam Cheshire, who has a lab in Utah now, and we're continuing the collaboration. I would like to really thank my colleagues at the Pediatric Neuro-Oncology, especially my lab, and I apologize. Oh, this is an old slide. I really apologize for this. My lab, which is Alison Cole, Senthil, Eric Hoffman, and Jocelyn Cruz, who are, I think, in the audience today, and I apologize to them for not having the name here. Rajiv Vivakar and Nick Foreman, who have been excellent mentors, as well as Leah Gore, who has supported the work which has been done in the lab, and especially the Morgan Adams Foundation. And at this point, I will start reading out the questions.
Great, thanks, Sid. You can go ahead and read the questions, and then if anything gets past you, I'll jump in and let you know. We have about 15 minutes left.
Okay. So Payal asks whether if I think CAR NK cells may have a role to play in tumors such as DIPG and glioblastoma. Actually, Payal, that's a very, very good question, especially because if you see, there were these two fabulous papers. One came out of Michel Mange's lab and one from Nick Vitanza from Seattle. And one of the things that they both put out, I think it was in Nick Vitanza's paper actually, which really talks about which really talked about NKGD, I believe, which makes the cells, which suggests that NK cells actually may have an activity in these models as well, especially DIPG and GPM. So the answer to your question is that this is probably something which would work. And the second question is from Todd. Todd, yes, the signaling domains are actually, very surprisingly, I must say, the same that is used in CAR T cells. So this one actually has the same CD3 zeta, which has a very has the same signaling pathways as well as sequence, a very similar sequence to the Fc gamma R2A receptor. So it actually starts, it has the same signaling mechanism or activates it in the same way. I was very surprised to see how well it actually does work, even though it's just a CD3 zeta. And those are the only two questions here.
You have other questions out there that people haven't posted yet? Give it a second, I think. Oh, there we go. Oh, feel free to ask less questions here. Guess you had a very thorough presentation.
Or I was too fast.
No, I don't think you were too fast. Any other questions? So I guess I had a question, and you may have explained this scientifically, but to my layman's brain, I missed it. Do you think this has specifically better applications for medulloblastoma and/or brain tumors than other types of solid tumors, or is that just your expertise, so that's why you're applying it?
So this is just my expertise. I mean, in fact, some of the best data has come from non-CNS tumors. They have done some clinical trials with, they have done some, I must say, successful clinical trials with hematopoietic tumors or liquid tumors. They have also done with myelodysplastic syndrome, and I believe there are ongoing trials with lung cancer. So this is a very common mechanism. It's highly conserved. It's a very common mechanism. So it's being done very rapidly in multiple different tumor types. Okay. But here, my expertise is of course focused within the pediatric brain tumors.
Okay.